Welcome back. This video is going to cover the basics of red blood cell morphology. Once you perfect the art of making a blood smear, it will have a nice teardrop shape, and it'll have three parts. It'll have the feathered edge where most of the white blood cells have fallen due to their weight, and we'll have everything kind of crowded together there. And then you'll have the agglutinated body, which is the majority of the blood smear. And in between all of that, where the feathered edge is kind of spaced out and the body is very congested, you have the monolayer, where the cells are spaced out in what is a normal arrangement for that patient. So make sure when you are observing cell morphology that you are inside of that monolayer section. The 5 ways we describe red blood cells are their size, shape, color, arrangement, and inclusions. When we're looking at the size, if a cell is a normal size for that species, we say that it's normocytic. If it's smaller than normal, we call it microcytic, and if it's larger than normal, we call it macrocytic. When there's a change in size in red blood cells, that is called anisocytosis. When there's a change in shape of red blood cells, we call that poikilocytosis. And there are many different forms of poikilocytes, and those include echinocytes, acanthocytes, target cells, schistocytes, keratocytes, dacrocytes, and ovocytes. And there's plenty more. And they're all related to certain kinds of conditions or pathologies. However, they're only pieces of a puzzle. For example, target cell may be related to different types of liver conditions. When we're looking at color, we say that it's normochromic if it's normal. If there's not a lot of hemoglobin and possibly the patient's anemic, the cell may be pale. We call it hypochromasia. And when there's a lot of color, we call it hyperchromasia. If there's a lot of different colors going on in that blood, we call that polychromasia. So there's several different colors we're seeing. When the arrangement is normal, the cells are spaced out from each other in the monolayer. Sometimes we may see overlapping of cells, which is called rouleau and is common in cats and horses. And we also may see agglutination, which is not normal in the monolayer. Other things we may see inside of a red blood cell are inclusions. These are in the red blood cell, like intracellular parasites, residual DNA or RNA from immature red blood cells, or nucleated red blood cells, which still have the full nucleus. And we also may see bacteria or something like a Cabot's ring from different parasites. And that is it. That is the 4 components of blood. If you want to know more, check out some of our other videos. And don't forget to check out our website for some cool vet tech products.